This video looks at what happens if we have other sources of energy. So far we've looked at glucose, but what about other molecules that we can use for energy? And then it also looks at how we can regulate cellular respiration. How can we turn it on? How can we speed it up? How can we slow it down or turn it off? So that's what we'll take a look at here. We do know that there are other molecules that can be used to go into this energy producing pathway. Some can connect to glycolysis, some can connect to the citric acid cycle. This figure gives us a brief overview of how that might occur. So we could take other carbohydrates other than glucose, some polysaccharides like starch. We could break those down into their glucose units, and then we could perform glycolysis on those. We can also use some amino acids that we can ship into glycolysis. We can also take glycerol off of our triglycerides and we can send those into glycolysis. Some amino acids can enter that pyruvate oxidation stage, and then some amino acids can enter the citric acid cycle. Some fatty acids can enter the citric acid cycle. So we can use other energy sources other than glucose to create ATP. This shows us a little bit more. We can take our proteins, break them down into their amino acids, and then some of those can be converted to acetyl-CoA and other things we'll see in a few moments. Our polysaccharides could be broken down into their monosaccharide units and then converted to acetyl-CoA just like we did through glycolysis. Fats can be broken down into their fatty acid chains, and then those fatty acid chains can be broken apart to form acetyl-CoA. And then all of those could eventually go into the citric acid cycle and run through oxidative phosphorylation. So we have a variety of different molecules that we can use to extract energy. Recall what an amino acid looks like. It had the central carbon, it had a carboxyl group, it had the amino group, and then it also had this R group, which varies from one amino acid to another. Exactly what happens to amino acids will very much depend on what this R group looks like. But we do know that, at least for here, we have a 2-carbon unit right here that we could create acetyl-CoA with, because remember, acetyl-CoA is a 2-carbon unit, and then we could run that through the citric acid cycle. The first thing we would have to do, though, with an amino acid is to get rid of this amino group. And then we can use the rest of this for energy production. Over here we see a triglyceride. In this case, what we typically will do is we'll take off this glycerol molecule and use that. And then we also will take these fatty acid chains and break them into 2-carbon units, which is what is happening right here, because once again, remember, this is a 2-carbon compound. So we can break this chain off, break 2 carbons off, and create acetyl-CoA and run those through here. This step is beta oxidation. This gives us a little bit bigger picture of what we can do with some of our different energy sources. This again is saying it will take our proteins and break them down into their amino acids. Obviously, we want to get rid of nitrogen, but if we're looking at how it's going to generate energy, we can take these amino acids, convert some of them to acetyl-CoA. We can run those through the citric acid cycle. Various carbohydrates can be broken down into glucose, which then becomes glucose 6-phosphate, and then that can go also into this normal pathway we saw before, pyruvic acid to acetyl-CoA to the citric acid cycle. Another thing that this is showing us is what might happen if we have more energy than we need. Then one of the things we could do is we could actually do this in reverse. We could do gluconeogenesis. We could reform glucose 6-phosphate, and then we could reform some glycogen if we have excess. Glycogen is a storage carbohydrate, and you see a lot of glycogen in like your liver and muscle cells, and it's there for breakdown of energy. So if we need to use energy in that cell, we can break glycogen down into its individual glucose units and run the pathway this way. But if we end up with excess, we can always go the other way and make glycogen. This is showing us the breakdown of the lipids. So we would take our lipids, we would split off the glycerol molecule and do a beta oxidation, and we also can take those fatty acids. After we do the beta oxidation, they become acetyl-CoA, and we can run them through the citric acid cycle. This just shows us some of those various pathways and how they interconnect with one another. Now, the energy gain for these different pathways will vary. We saw that if we started with glucose and we went all the way through glucose to pyruvic acid to acetyl-CoA, through the citric acid cycle and down the electron transport chain, we got approximately, say, about 32 ATPs. If we use different pathways, we will get a different amount of energy. Let's take a look at protein metabolism. The amino acids that enter the citric acid cycle are going to do it at different places because it's going to depend on the amino acid. That amino acid has the central carbon, the amino group, which we're going to strip off, the carboxyl group, then there's that R group. And that R group varies from one amino acid to the other. So depending on what the R group is, these amino acids can enter at very different places. So this gives us an idea of where some of these amino acids would enter. You don't need to know the details of this, just to show that there is a variety of different places these amino acids can come in. So these amino acids might be converted to pyruvate, and likewise pyruvate can be used to generate these amino acids. And then some might be sent directly into acetyl-CoA. Others might enter in different places along the pathway. So you can see with the proteins, depending on the amino acid you're burning, you might get a very different energy output. Because if there are some ATP generating steps further up and we enter later in the pathway, then we're not getting those particular ATP that were formed during those steps. Now let's take a look at how we regulate cellular respiration. How do we make sure that we are producing enough ATP, but not too much, and providing the cell with the energy it needs, but not being inefficient? Because remember, if we produce the ATP and we don't use it, it's an unstable compound. It'll just break back down. And then we've just lost the energy that we've put in. So that's an inefficient way for a cell to work. So how do we keep that from happening? There's a lot of different regulating mechanisms that control this because it is something that throughout history would have been under very strong selection for cells to be as efficient as possible. One is we can control hormonally how much glucose actually gets into the cell. If a cell needs energy, then we can increase the rate at which glucose enters the cell, and if it doesn't, we could possibly decrease the rate at which glucose would enter that cell. We also have enzymes that are reversible. Some enzymes, if they're reversible, they can essentially sort of turn off temporarily when the substrate is very low and the product is very high. If that's the case, if we have a lot of product and not a lot of substrate, then we're going to turn our factory down a little bit or maybe even turn it off temporarily. Enzymes also are going to be sensitive to changes in pH. Because they're proteins, they're very sensitive to pH changes. In some cells, if the pH changes due to lactic acid, that could affect the sensitivity of those enzymes and make them function at slightly different rates. And then we have some feedback controls. And that's what we want to look at next. Feedback loops in metabolic pathways that allow them to be under control. One of the regulatory mechanisms we want to think about is the idea of getting the glucose into the cell. This was that first one we talked about, the hormonal control. So in this hormonal control, if this cell is, let's say, low in glucose, then the cells would essentially cause the body to secrete more insulin, and that insulin would go and bind to these receptors, and then those receptors binding to the insulin open some of the GLUT transporters in the membrane, allowing sugar to get into the cell. We can also do it through this enzymatic means that we briefly mentioned, where we can take products of these different reactions and use those products to inhibit some steps in the pathway, or we could actually take the reactants and use those to stimulate. So if I have a lot of reactant and very little product, I want to turn this machinery on so I can activate it. If I have a lot of product, so a lot of ATP, then I want to turn this off. I don't need to run it. ATP, as you're seeing here, can come back up and inhibit this pathway. And so can some of the molecules that are generated from that citric acid cycle can also come back up and inhibit it. We'll take a look at some of this regulation in a little bit more detail. Here's a summary of it, and then we're going to look at each one of these a little bit more. But this just gives you one place to look at sort of some of the different ones. So over here we've got the different pathways. Glycolysis steps can be regulated. This pyruvate oxidation can be regulated. The citric acid cycle and the electron transport chain. We'll take a look at each of these. Let's look at the control of glycolysis. One of the early enzymes in glycolysis is this hexokinase. We see it here. Now, what would we think would want to turn off hexokinase? This is the product of that reaction. Just look at these reactions right here. This is a product. If I get too much of this, I could turn this off. And that's what we see here. If glucose 6-phosphate increases, we decrease the activity of that enzyme. Let's take a look at another very important enzyme in this pathway. This is a major point of regulation of glycolysis, phosphofructokinase. What is the product of phosphofructokinase? One is the fructose 1,6-bisphosphate. Imagine this going a little bit further and entering the citric acid cycle. ATP can come back and decrease the activity of this. If fructose 6-phosphate is high in the cell, then it will increase the activity of this enzyme so that we can run the reaction this way a little bit more. So if this goes up, the reaction shifts to the right. Think of Le Chatelier's principle from chemistry. If citrate, which was over in our citric acid cycle, is high, then this is going to slow this down a little bit. And also if it's acidic. So if pH starts to drop a little bit, that will also tend to slow down the phosphofructokinase. We also have the pyruvate kinase. If we look at pyruvate kinase, it's way down here. If you think about what types of things could possibly slow down this enzyme, remember the step that comes right after this pyruvate is the formation of acetyl-CoA. If acetyl-CoA gets high, it's going to come back and slow this down. And then, of course, ATP as well, because all along this pathway, the ultimate end result is ATP. Typically, high ATP levels have the ability to come and influence a lot of these different enzymes on this pathway and slow them down. Here we'll see a video that shows us this regulation of glycolysis.
In this lesson, we'll discuss 3 ways on how glycolysis is regulated. The question reads, how is glycolysis regulated by each of the following enzymes? Starting with the first, we have hexokinase. It's important to note that glycolysis is a 10-step reaction that produces cellular energy from glucose in the absence of oxygen. Throughout the process, which happens to take place in the cytosol of the cell, it degrades the 6-carbon glucose molecule to 2 3-carbon pyruvate molecules. In the very first reaction of glycolysis, phosphorylation takes place. This is where a phosphate group is transferred from ATP to glucose, forming glucose 6-phosphate. Also in this process, the dephosphorylation of ATP forms one molecule of ADP. The enzyme hexokinase is responsible for this reaction, and it is inhibited by high levels of glucose 6-phosphate. So when a lot of glucose 6-phosphate is formed, this eventually causes a negative feedback loop to stop the addition of more phosphate groups being added to glucose. Therefore, what stops hexokinase is glucose 6-phosphate. In the third reaction of glycolysis, another phosphorylation reaction takes place. This time, phosphate from ATP is transferred to fructose 6-phosphate, producing fructose 1,6-bisphosphate. As the name suggests, this molecule contains 2 phosphate groups. A second kinase enzyme called phosphofructokinase catalyzes this step through hydrolysis. This enzyme is inhibited by high levels of ATP and activated by high levels of ADP and AMP, an indication that energy needs to be replenished in the cell. Finally, for C, pyruvate kinase. In reaction 10, which is the last reaction in glycolysis, phosphate groups from 2 phosphoenol pyruvate molecules are transferred by pyruvate kinase to produce 2 pyruvates and 2 ATP molecules. High levels of ATP or acetyl-CoA, which is a molecule that's produced from pyruvate, inhibit pyruvate kinase, and this stops the formation of more pyruvate being formed. And there you have it, a quick discussion on how glycolysis is regulated.
Next, we have control of the next step in the pathway, which is pyruvate oxidation. Remember, we're going to convert pyruvate to acetyl-CoA, and then we're going to ship acetyl-CoA into the citric acid cycle. This step can be regulated by the enzyme that drives it here, pyruvate dehydrogenase. Take a look at the product of this stage is acetyl-CoA. So if acetyl-CoA builds up in the cell, it can come back and do a feedback and turn this enzyme off. So acetyl-CoA, high levels of acetyl-CoA will cause this reaction to decrease. We're also forming ATP along this entire pathway, and in this step, we've also created NADH. Both of those things, which are both products, can also decrease the activity. What could increase the activity? ADP, because remember, if the cell needs energy, ADP is going to be high and ATP is going to be low. If ADP is high, the cell needs energy and it turns this factory on so that we make more ATP. The reactant in this reaction is pyruvate. So if I have a lot of pyruvate, it's going to increase this reaction rate. Now let's take a look at control of the citric acid cycle. So you see two major enzymes being controlled here, isocitrate dehydrogenase and ketoglutarate dehydrogenase. The isocitrate dehydrogenase enzyme is used in step 3 to convert isocitrate to alpha-ketoglutarate. We know that throughout this process we are creating ATP. We're also creating NADH. ATP and NADH, since they are products, if those levels get high, it will decrease the activity of this enzyme. Since ADP is a reactant in this, then high levels of ADP would increase the activity of isocitrate dehydrogenase. The next enzyme we have here, the ketoglutarate dehydrogenase, we see here in step 4, going from alpha-ketoglutarate to succinyl-CoA. So what are the products here? Well, succinyl-CoA is one of our products, and if that level gets high, it decreases the activity of this enzyme. We also have as products ATP and NADH like we had before. If those levels are high, will decrease the activity of the enzyme. ADP is a reactant, therefore if it's in high quantity, it will increase the activity. Here we'll take a look at a video that talks about some of the consequences of mutations and malfunction in isocitrate dehydrogenase.
Cellular metabolism is essential for all cells to carry out their functions, and altered metabolism plays an important role in the development of many cancers. In normal cells, IDH enzymes facilitate the cell's energy production from glucose and other metabolites. IDH1 localizes to the cytoplasm, IDH2 to the mitochondria. IDH1 and IDH2 enzymes catalyze the conversion of isocitrate to alpha-ketoglutarate. This reaction requires NADP as a cofactor, which is converted to NADPH that can help protect cells from the toxic effects of reactive oxygen species. In addition to its role in metabolism, alpha-ketoglutarate is an important substrate for cytoplasmic and nuclear alpha-ketoglutarate-dependent enzymes to carry out their normal functions. In the nucleus, these enzymes help regulate DNA and histone methylation patterns, which turn the expression of genes on and off, including genes critical to cellular differentiation. In IDH mutant-positive cancers, IDH mutant enzymes acquire a new or neoenzymatic gain-of-function activity that may disrupt normal cellular metabolism. The IDH mutant enzyme dimer has a normal and a mutated copy. The normal copy generates alpha-ketoglutarate. However, the mutated copy takes alpha-ketoglutarate and converts it to the oncometabolite 2-hydroxyglutarate, or 2-HG. This reaction consumes NADPH, which may increase reactive oxygen species and thus promotes oncogenesis. This leads to an accumulation of 2-HG levels within the cell. The excess production of 2-HG can disrupt the function of cytoplasmic and nuclear alpha-ketoglutarate-dependent enzymes. This disruption occurs because of the chemical similarity between 2-HG and alpha-ketoglutarate. 2-HG binding to alpha-ketoglutarate-dependent enzymes prevents their ability to demethylate DNA and histones. Therefore, elevated levels of 2-HG are associated with the abnormal methylation of DNA and histone proteins. Research suggests that hypermethylation of DNA and histones alter the transcription of key genes, including those necessary for differentiation. The result is a block in cellular differentiation and an accumulation of immature cells, which potentially contributes to oncogenesis in IDH mutant cancers. Inhibition of the IDH mutant enzyme prevents 2-HG production and is believed to help restore cellular metabolism. The decrease in 2-HG levels allows alpha-ketoglutarate to bind to alpha-ketoglutarate-dependent enzymes and may restore function of DNA and histone-modifying enzymes. This can result in reversal of DNA and histone hypermethylation and allows expression of genes that are involved in cellular differentiation. Preclinical studies suggest that inhibition of IDH mutation releases the differentiation block, which may allow immature cells to differentiate rather than persist and grow as a tumor. IDH mutation inhibition may have important implications for cancer cells with IDH mutations, and investigations are underway to determine their clinical potential.
Finally, we'll take a look at the control of the last part of cellular respiration, which is electron transport. We know that electron transport is responsible for producing most of the ATP in cellular respiration, and of course it uses ADP to do that. So ADP is a reactant and ATP is a product. So it makes sense that as we build up ATP in the cell, it will tend to decrease the enzymes that work in the electron transport chain. And if ADP builds up, it will increase the activity of those enzymes in that pathway. So the general idea is that as products build up, we tend to decrease enzyme activity, and as reactants build up, we tend to increase activity. That completes our section on the control of cellular respiration.